Yes, yes, sir.
Okay, good. So, in this class, we are going to learn about a few concepts in the clinical research area. So, before we start with some specific terms in the clinical research, first we'll try to learn what is clinical research in reality. Okay. So there are many definitions of clinical research. One definition that I find that this is the profound definition of the clinical research, and this is clinical research is the study of health and illness in people. Okay, it is the way we learn how to prevent, diagnose, and treat illness. So we are covering all the things, right? We are covering the prevention, we are covering the diagnosis, and we are covering the treatment. So now we know what is clinical research. It is nothing but a study of health and illness in people. And the main purpose of doing the clinical research is to identify the ways to prevent, diagnose, and treat the illness. Now, how many types of clinical research are there? So, as for the FDA, these are the different type of clinical research. So, this is not a very standard classification of clinical research, but these classifications can summarize many type of clinical research activities in a real world. Okay, so the first of them is treatment research. Okay, so with the name itself you can understand what is treatment research. The name itself is telling that we are doing something with the treatment. Okay, so in the treatment clinical research, what we do, we try to identify new treatment. And the second one is prevention research. Now again, with the name itself you can understand, right? Anything which is being done for the prevention purpose. So in the prevention research, we'll try to identify the ways to prevent any infection. For example, if a person is suffering from pneumonia, then we'll try to identify the chemicals which are drug which can prevent the secondary infection. For example, he should not, if he is having pneumonia, then he should not be having syphilis or something like that. Okay. Then third one is diagnostic research. Again, in the diagnostic research, we'll try to identify various ways to diagnose any particular disease. Screening research. In screening research, we'll try to identify various ways to screen the subjects. For example, various ways to screen cervical cancers in women. Okay, quality of life research. In this type of research, what we do, we try to identify the ways to improve the quality of life. For example, if patient is suffering from terminal illnesses, means there is no cure for that particular illness, and he is in very much pain, then we'll try to identify the ways to age his or her pain. Okay, to minimize the pain, so his or her quality of life can increase. Then next one is epidemiological studies. So epidemiological studies means we try to identify the pattern of a disease. For example, recently we have seen COVID, right? So whatever research was done related to the COVID to identify the pattern, how it spreads, how people get in COVID infection and all, those kind of research comes under epidemiological studies. And last one is genetic studies. So with the name itself, you can understand we are trying to identify the genetic link with the disease. So these are the different type of uh, clinical research studies that are performed in the real world. So many studies may be common, right? Some studies might be the screening research, that but the same research may also fall under diagnostic research category as well. So this classification I have shared with you to give a general overview about the clinical research. Now, in today's class, we'll focus on the first one, the treatment research. So, as I said, in the treatment research, we try to identify the various treatment, okay, for the treatment of a particular illness. So, now how we determine that particular treatment or particular medication or particular device or any diagnostic procedure or surgical procedures are safe and effective. Okay, so how we are going to determine it? So in the next slides, we'll try to understand the proper method to determine the safety and efficacy of medication, device, diagnostic procedure, or surgical procedures. Okay. So to determine the safety and efficacy of any treatment, we run the experiment. Those experiments are called clinical trial. So people talk about clinical trial. So now what is clinical trial? So clinical trial are the experiment which are designed to answer a specific question. In what that specific question is telling whether new drug or device or procedure is safe and effective or not. Simple. Okay. So here we understand that clinical trial are nothing but experiment which we do to determine that our drug or any medical devices or any procedure. When I say procedure means any surgical procedures, okay, which is safe and effective or not. So whole purpose of clinical trial to determine these two things only, the safety and effectiveness. Okay. So clinical trial is a part of long drug discovery process, which may take many years to complete. So the drug research does not start with the clinical trial. It starts from basic. Now we'll try to understand what is that basic. Okay. So in the drug developmental stages, the clinical stage comes in last. Okay. It's the third part. The clinical the the drug discovery process starts with the discovery. So we'll try to understand what is discovery in various part of the discovery, then various part of the preclinical stage, and then various stages of the clinical part of this long discovery process. Okay. So starting with the discovery stage of development. So again, the discovery stage of development can be divided into three parts. Okay, 1 minute. So as I said, discovery stage of development can be divided into three parts: drug discovery, target validation, lead generation, and refinement. Now we'll understand what they are. So in target discovery, what we do simply first we need to identify the nature of the disease, right? We need to identify a particular protein or particular gene or particular receptor which is causing that particular illness. Then only we can make medicines, right? So in the target discovery, what we do? We try to identify any proteins or any gene or any receptor which is causing that particular illness. So whenever any sponsor company, any pharma company want to develop any drug molecule, so they always try to identify the target which meets its clinical and commercial requirement. For example, malaria, or for example, congestive heart failure. Okay, so there are many ways to prevent that congestive heart failure, right? Lowering of the cholesterol, controlling of blood glucose, or maintenance of the blood pressure. So but there are many drugs which are targeting these things. So any pharmaceutical company will try to identify that particular molecule which is not yet been discovered, or which is not yet known, or there is no drug available which is targeting that particular protein. So they will try to know more about that particular target. So whenever a sponsor company starts with the discovery stage of development, they try to identify the target which meet their commercial requirement as well. Okay, so in nutshell, in the target discovery, we'll try to identify the particular thing which is causing a disease. Okay, once we know, okay, these are the protein or gene which is causing that particular illness, then what do we do? In target validation, with the name itself, we can understand validation. In second stage, we are going to validate it. Okay, in the target discovery, we have that primary information. Okay, in target validation, we will further confirm we'll confirm whether whatever we have identified in the target discovery is a good target or not. So now how we will do it? We'll do it with the help of some animal or cellular model. Okay. So we'll manipulate a particular gene. For example, we have identified that gene A is causing that particular illness. So we'll try to inhibit or overexpress that particular gene, and we'll see whether it is causing that particular disease or not. Okay. So we create many animal and cellular models to confirm the target. Now, third stage is lead generation and refinement. So in lead generation and refinement, we try to create many molecules, okay, which is targeting this target. Okay. So there are many, like you would have heard this word, high throughput screening. Means there are many robotic processes where we screen thousands of compounds in a single go. Like in few minutes only we can screen thousands of compounds. So by using various robotic methods or various new technology, we screen thousands of molecules with respect to that particular target that we have identified in these two steps, in target discovery and target validation. And once we identified few molecules, say example 100 or 50 molecules we identified, then we further test those molecules. We add some group like uh, um, methane group, sulfur group. We try to add or remove a particular group from that chemical moiety or chemical compound, and try to increase the binding with respect to the target. Okay. Or we want to increase the solubility, or we want to increase the absorption. So we'll try to do everything in order to refine that particular molecule. So this process is called lead generation and refinement. And after identification of a particular lead molecule, the second stage starts, that is preclinical stage of development. So in the discovery stage of development, we have identified the target, we have validated the target, we have created the molecules, and we have identified the best molecule. Okay. With that best molecule, we enter in the preclinical stage of development. In the preclinical stage of development, what happens? We test that particular molecule in animal models. And for what we test? In order to determine the efficacy, toxicity, and PK, pharmacokinetic. Okay. So pharmacokinetic means its absorption, distribution, metabolism, and excretion. To determine these four qualities, toxicity means whether it's having any toxic effect along with its efficacy or not. Okay. So we always try to have a balance between these two. It is less likely that any molecule will not have any toxicity. So always in the preclinical stage of development, we try to have a balance between efficacy and toxicity. The drug should be more efficacious and it should be least toxic. Okay. And what do you mean by efficacy? Efficacy means how successful is the performance of a drug candidate. For example, if we are testing anti-tumor drug, then how well it is going to shrink the tumor. Okay, that characteristic, that how well it is going to shrink the tumor, is called efficacy. Okay. And in the preclinical stage of development, we follow one guideline that is called GLP, good laboratory practices. Okay. Now we learn about two things: discovery and preclinical stage of development. Okay. Now the drug molecules will enter into the clinical stage of development. So again,
Hello.
the yeah.
Sorry, sir, disturbing. I think your slide is stuck. Slides
Okay.
are not
And
moving. Only first slide are there.
Only first slide.
Now the are not moving.
Okay, I got you. Okay, let me reshare it again.
Yes, sure, sir.
Okay, please let me know if you can see it. Please do let me know if you can see my screen now. Am I audible?
Is there audible?
Yes,
But
sir.
it's not visible. Screen is not visible properly.
Okay, I think there's some technical issue. Now also it's not visible.
But it's not visible.
Okay. I reshared it. Now please let me know if you can see it. Hi all, please let me know if you can see my screen.
Yes, sir. Now it's visible.
Like I'm moving my slide, it's visible, right?
Yes, yeah.
Yes, yes,
Okay, great.
yes.
Uh, okay. I'm sorry for this technical glitch. I'll just summarize what I've seen because you people would have heard what I said, but you people would not have see my slide. So I quickly summarize what I said. Like I just completed 3 or 4 slide only. So I started with the definition of clinical research, where I said about the basic definition of clinical research, which is nothing but the study of health and illness in people. Then I summarized about the different type of clinical research. I started with the treatment research and I end with the genetic studies. And out of these studies, I selected treatment research for today's class. So then I said, like, what is the purpose of treatment research? And then it is nothing but to determine the safety and effectiveness of a medication, device, diagnostic procedure, and surgical procedure. Okay. So how we do this? So we run the experiment to determine the safety and efficacy of a treatment. Now those experiments only called clinical trial. So in clinical trial, we try to answer a specific question to determine the safety and efficacy of a particular treatment. Treatment can be any drug, any medical device, or any procedure, means any surgical things. Okay. So we don't start directly with the clinical trial. We start with the basic. So what is that basic? So to understand that basic, we need to understand the drug developmental stages. Okay. Then I said about various drug developmental stages: discovery, preclinical, and clinical stage. So we already completed about the discovery and preclinical. In discovery, I said the discovery stages can be divided into three part: drug discovery, then target validation, then lead generation and refinement. In target discovery, I said that in target discovery, we try to identify the protein, gene, receptor, which is causing that particular illness. Okay. In target validation, what do? We confirm whatever we have identified in target discovery, we confirm here. In the third stage is lead generation and refinement. What do we do here? We have identified the target, we have confirmed here in target validation. In lead generation and refinement, we try to create different molecules. Then we modified that molecule and we try to identify the best molecule. After identification of that best molecule, the second stage, which is preclinical stage of development, starts. What we do here? We test that best molecule that we have identified in previous step. We test that particular molecule in animals. Okay. For what purpose? To determine the efficacy, to determine the toxicity, and to determine the pharmacokinetic. Now, what do you mean by efficacy? Efficacy is nothing but it tells how successful is the performance of the drug candidate. For example, if we are making an anti-tumor drug, then how well that the drug is going to shrink that particular tumor is is called the efficacy of that particular drug. Okay. And toxicity, you people all understand, right? Toxicity means whether it's having any negative effect on the cells, organelles, or any pathophysiology of the body. And pharmacokinetic means four properties: absorption, distribution, metabolism, excretion. Okay. So we learned so far. Now we have to start with the clinical stage of development. So again, the clinical stage of development can again be divided into four steps: phase 1 step, phase 2 step, phase 3 step, and phase 4 step. Okay. We'll start with the phase 1. Okay. So so far we have learned that in order to initiate the clinical trial, we need to complete two steps: discovery and preclinical. Again, in discovery, we have three steps: target discovery, target validation, and lead generation and refinement. Then preclinical stage, then clinical trial. So the clinical stage of the development start with phase 1. Okay. Now we'll learn what is phase one. So the phase one is nothing but first in human trial. Why it is in first in human trial? Because after the preclinical stage, this is the first time we are going to test the drug in humans. So in the clinical stage of development, we test the drug in humans. Okay, and phase one is the first step. In how many people we test the drug? Generally, it is tested in 20 to 80 people. Okay, this number may vary. Okay, some books may suggest like 30 to 70 or something like that, but generally it is less than 100. Okay, and what we determine in phase one, we test safety, PK, and what else? Maximum tolerated dose. Now, what is safety? Everybody knows what is safety because it's a general term, right? Safety means whether it is harmful to the human body or not. Then PK means absorption, distribution, metabolism, and excretion. Then third is MTD, maximum tolerated dose. So maximum tolerated dose is nothing but it's telling how much dose a person can tolerate. Okay. It So again, now we with the help of example we understand how these things are determined. Okay, but for now, just understand that we determine at least these three things: safety, that is first, then second is PK, and third is maximum tolerated dose. Okay, so again, the phase one is further divided into two types: phase 1A and phase 1B. Phase 1A is called single ascending dose study, and phase 1B is called multiple dose ascending study. Generally, people will understand phase one, then phase two, then phase three, but every step is not that simple. Okay, it is having many further baby steps. So we have to learn each step. So phase one is divided into two parts: phase 1A and phase 1B. Phase 1A is called single ascending dose study, and phase 1 is called multiple dose ascending study. We try to learn with the help of example. Okay, so in phase 1A, what happens? A small group of subjects are given a single dose of the drug. Okay, for example, they select 30 people, they give 30 mg of a drug, and they observe for 7 days, and they take the blood and perform their assessment. Okay, so how we determine the maximum tolerated dose? For example, they have taken 30 people. Okay, and they have given the drug, and for after 7 days they have checked after how many people have abnormal liver parameter, LF, AST, ALT. Okay, based on that they will determine, okay, now 10 people are suffering from abnormal liver parameters, means this should be our maximum tolerated dose. Okay, in phase 1B, a group of patients receive multiple low dose of the drug. Here all people are receiving single dose of a drug. Okay, just remember, here people are receiving multiple dose of a drug. Again, as in single dose, we are observing for some time and then we are testing. We are taking the sample from the subject and we are testing for safety parameters, right? Here also we will do the same thing. The only difference is here single dose of a drug is given, here multiple dose of a drug is given. Okay, we'll see one live example. For example, for phase 1A, three subject. I have just chosen three subject. However, in phase one, we choose right 20 to 80 subject. Just for simplification, I have chosen here three subject. Okay, so we are giving here 50 mg of a drug. We have seen, okay, there is no adverse effect on the subject body. Whenever I am seeing subject, it is nothing but patients. Okay, again, three more subjects are recruited, and we have increased the drug. That's why it is ascending. You see, a ascending word is coming, right? We are increasing the drug here. Now we have seen that one subject is having unacceptable toxicity. Okay, so three out of one, it means one third, right? Now it is one third. So again, we will record further three subjects and we'll give the same drug, and again we'll see whether it's having unacceptable toxicity or not. If one subject or more than one subject is suffering from unacceptable toxicity, we will say, okay, this drug is maximum tolerated drug. Okay, we cannot give any human being 100 mg of drug A. Okay, for example, you would have heard, right, that paracetamol cannot be taken like 8 gram in 24, more than 8 gram in 24 hour, for 24 hours, right? That 8 gram is the maximum tolerated dose for the paracetamol. And in this type of experiment only we'll determine those kind of doses. Okay, this is a simple example of single ascending doses study. Now we have seen the example of phase 1A. Now we'll see second example for the phase 1B. So as I said, in phase 1B, we are giving multiple dose, right? You see here, we have recruited multiple subjects in different group and we are giving different drug. Like in week 1, we are giving 50 mg, then 60 mg. In group B, we started with 60, then group C, we started with 70, then in group D, we started with 8, and then we are increasing it. Okay, that's why it is a multiple ascending dose study, because we are giving different type of different doses of the drug in a ascending order. Okay, again we'll do the same thing. We'll take the blood sample and we'll see whether any subjects are having abnormal lab parameters or abnormal ACG or anything else. Okay, now everybody is clear, right, about phase 1A and phase 1B. If anybody is having any question, then please let me know before we proceed with the phase 2. Okay, so the key takeaway from phase 1 is it is done in healthy volunteer. Okay, because this is the first time we are going to test the drug. Okay, we cannot take the patient with multiple illness, we have to take the healthy volunteer. But there are few exceptions. For example, in cancer trials where terminally ill patients are there. Okay, and those kind of uh, trials where very toxic drugs are tested, we generally prefer to use patients instead of healthy volunteer. But whenever somebody is asking you in phase 1 which type of volunteer, which type of subjects are recruited, you have to say like healthy volunteers are only recruited. But there are few exceptions if trial is being done on a terminally ill subjects. For example, in an oncology studies, uh, instead of healthy volunteer, patients are recruited because drugs are generally cytotoxic. Okay, and how many people are recruited? As I said, 20 to 80 people. This number may differ, but it will be always less than 100. And what is the goal of phase 1? Mainly safety, then MTD, then PK. Okay, leave about PD for now. When we'll see in our normal classes, I'll explain more about the PD part. Okay, for now, the main goal of phase 1 is safety, MTD, maximum tolerated dose, and PK. And it has two parts, phase 1A and phase 1B. Phase 1A is also called single ascending dose study, and phase 1B is called multiple dose ascending study. And this part is very critical because generally whenever you are appearing for any job interview and you have to write the technical questions paper, then generally in multiple choice question they will ask this question. Okay, what is phase 1A and phase 1B? Now after phase 1, the drug is moved to phase 2. Okay, so what is phase 2? Phase 2 is also called proof of concept, POC. Always remember, okay? And it is also called non-pivotal trial. I'll tell you why we are using these two terms. Okay. And third thing is it is tested in large group of people, typically 100 to 300. In phase 1, what I said, less than 100, right? Here 100 to 300. And what we do in this phase 2, we determine efficacy and safety. In phase 1, what we did? We determine safety, PK, and maximum tolerated dose. We did not determine efficacy. Okay, just remember, this is the first time we are going to determine efficacy. That's why it is called proof of concept, where here we are going to prove our concept. Okay, my drug is effective for this particular illness. and it is non-pivotal. Okay, this is technical word. Whenever we are going to submit the data of any clinical trial to the regulatory authority, okay, to the regulatory body for taking approval, those kind of trials are called pivotal trial. Okay, so phase 2 trial data generally not submitted for taking approval, okay, for any drug molecule. Like based on the phase 2 data, any sponsor companies cannot sell the drug in the market. Okay, that's why it's called non-pivotal trial, and it is also called proof of concept trial. Okay, everybody is clear, right? Because this is the first time we are going to test the efficacy. Always remember this is very important. Now again, as in phase one, I said phase one is divided into two parts: phase 1A and phase 1B. Similarly, phase two is also divided into two parts: phase second A and phase second B. Now, what is phase second A? Phase second is called exploratory trial, and phase second B is called definite dose finding studies. Okay, so I'll tell you what we do in phase second A and phase second B with one simple example. For example, in phase one, we have determined that what is the maximum dose that person can tolerate, right? We have determined that. For example, 100 mg is the drug. 100 mg is the dose of a drug which can be tolerated by any subject. Then what we do? We give drug 10 to 100, and we try to see whether that dose is having any effect on illness. For example, if we are testing the anti-tumor drug, okay, then we will give that subject 10 mg, 20 mg, 30 mg, up to 100 mg. We cannot give more than 100 mg because in phase 1 we have determined this after MTD the drug will be very toxic. Okay, that's why it is a maximum tolerated dose, right? So in dose range exploration, what do we give? 10, 20, 30, 40, up to 100, and we try to see the effect. Okay, at 10 mg, 20 mg, what is the effect? Once we identified, okay, at 60 mg, 70 mg, 80 mg, drug is showing the maximum effect. Again, in phase second, what we do with that range 60, 70, 80 mg, again we'll recruit some subjects and we'll try to give that drug. Okay, and we'll try to determine the best dose which is showing the maximum efficacy. Okay, so it's very linear. Okay, in phase one we determine the maximum dose person can tolerate. After knowing that, we try to test the different dosing to determine the best drug which is showing effect. Okay, in phase second. Now once we know the best dose which is very efficacious, the drug is moved to phase 3. Phase 3 is done in large group of people, highest like 3,000, sometimes 10,000 also. Like the vaccine trial that we had in COVID, that was done in more than 10,000 people. Okay, so generally it is done between 1,000 to 3,000 or 4,000 people, and it is a final confirmation. Okay, for what? For safety and effectiveness. And why it is done? Now, minute side effects also we can determine here. We have to determine the drug-drug interaction, because in real world, okay, if person is taking the BP drug, then the person might be taking the diabetic drug as he might be having the asthma as well. So we have to determine the drug-drug interaction as well. We have to determine the drug-food interaction. A person might take the drug with the juices, might take the drug with the milk. So we have to determine the drug-food interaction also. And the last thing is we have to compare it with commonly used treatment. For example, uh, uh, we are developing a drug for BP, but are for cholesterol. Okay, atorvastatin is a drug which is used for to lower the cholesterol level in the blood, right? And we are testing our drug, so we have to determine whether our drug is good compared to the already existing treatment or not. That kind of comparison with respect to the standard treatment which is available in the market, that comparison is also done in phase 3. Okay, and phase 3 data is submitted to the regulatory authority. Okay, and that's why this phase 3 trial is called pivotal trial. Phase 2 trial is called non-pivotal trial because data is not submitted to the regulatory authority for taking permission for the sell in the market. But phase 3 data is submitted to the regulatory authority seeking approval to sell the drug in the market. That's why phase 3 is called pivotal trial. Always remember. And second thing, I use this word effectiveness here. But in phase 2, I use this word efficacy. Okay, this is very important. When both the terms like efficacy and effectiveness tells the same thing. What I said, efficacy means how well the drug is working for our disease, right? How successful is drug performance in treating that particular illness. That characteristic is called efficacy, right? Definition of effectiveness is also same. But generally, whenever we are talking about large group of people, instead of efficacy, we use this word effectiveness. So whenever you will hear any press conferences about any drug, right? Generally, you will find this word drug effectiveness, something like that. Okay, because whenever we are talking the efficacy in a large group of people, instead of efficacy word, we use effectiveness word. Always remember. Okay, but both word meaning is same. So in nutshell, in phase 1, we determine the maximum tolerated dose person can take. We determine the safety and PK. In phase 2, we determine safety and efficacy. In phase 3, we determine safety and effectiveness and other things. Okay, then data is submitted to the regulatory authority for approval purpose. Okay, based on the data, the regulatory authority will determine the risk and benefit ratio and will approve or not approve any drug molecule. Okay. Now, once drug is approved by the regulatory authority, what happens? The phase 4 starts. Okay, it is also called PMS studies, post-market surveillance studies. Okay, always remember it is called PMS study, phase 4, and this is done after drug is approved. And why it is done? To check the drug performance in the real world scenario. Okay, because the drug is tested in a less number of people, say like 1,000, 3,000. But once drug is approved, it is going to be used by lakhs of people or crores of people. So we have to determine the risk and benefit of drug on a long-term basis, and we have to determine the real side effect. So what happens? Any side effect which happens like once in 10,000 or once in 30,000 people, that cannot be determined in any clinical trial. Okay, always remember. So you would have heard this controversy like Roficoxib. It was withdrawn in 1998 and 2002 because drug was causing cardiovascular toxicity. Okay, and but it could not be identified in a clinical trial because these were the rare side effect. So we came to know about these side effects by doing these phase 4 studies. Okay, PMS studies. Now we learned about phase 1, then we have learned about phase 1A, phase 1B, phase 2A, phase 2B, phase 3, phase 4. There one different kind of study that is called phase 0. Okay, this is not that much common now, but still it is done. Okay, by many organizations. So it is called micro dosing studies. So generally, what happens? It is tested in 5 or 6 people, sometime 8, 9. Well, generally, the number of people in which a drug is tested will not be more than 10. And it is when we do this phase 1, we do this phase 1. Sorry, when we do this phase 0, we do this before phase 1 means discovery, preclinical, then phase 0, then phase 1 will start. Okay. So in this particular scenario, phase 0 would be our first in human trial. Okay, not phase 1. But whenever somebody is asking you what is first in human trial, then it should be phase 1 only, because phase 0 is not something which is done usually, okay, for all the drugs. So what happens in phase 0? Drug is administered at subtherapeutic dose. Now you will ask, what is subtherapeutic dose? So with the term itself, you can understand here. The drug, the dose which is not going to cause any therapeutic effect, means it's not going to cure anything. Then why we are giving that drug? Okay, that's a normal question, right? If it is not going to do anything in the body, then why we are going to give that drug? So as you know that in preclinical we are testing the drug in animal models, right? So any animal model is not close to the human body, right? They have different enzymes, they have different things compared to the human body. So what we do to have a good idea about the drug's PK property, how well it is going to absorb in the body, how well the body is going to metabolize the drug, how well the drug is going to distribute in the body, and how the drug is going to excrete it from the body. To determine this quality of a drug, it is better to test the drug in human body, but we cannot give the drug to the to more number of people, right? So it's good that we test the drug in like in 5 or 6 people, and we give the drug at subtherapeutic dose. So subject will not having any side effect also, and we can determine the PKPD also. So this is the good way to determine the actual PK, actual pharmacokinetic property of a drug before going ahead with the phase 1. Okay. So how we do? It's very simple. Like uh, we will attach the carbon 14, which is a radioisotope, right, with a drug molecule, and we will inject that drug at subtherapeutic dose, means very minimal. Like in the animal model, we identify that 100 mg is the drug which may show some positive effect in human body for treating a particular illness. Then we will give only 1 mg. Okay, and we'll attach this carbon 14 isotope. Then with the help of some analytical instrument, we will determine the distribution of the drug in the body. Then we will take the blood sample, urine sample, and all. Then we'll see how well the drug is metabolized, or how well the drug is going to be excreted by the body. Okay, by doing this, we can determine the PK parameter very well compared to the preclinical models. Okay. Any questions so far? Like everybody is clear what is phase 0 and whatever we have learned so far? If no question, then I can initiate the next topic. The primary objective and the secondary objective. This is very critical. Okay. So what is primary objective? Again, primary whenever we have to determine anything in our work, we we always set the objective, right? We always set the goal. The same thing applies in the clinical trial as well. So primary objective or secondary objectives are nothing but they are the goal or aim, why we are doing that particular trial. Okay, so the standard definition of primary objective is, primary objective of the trial is to address the scientific question by collecting appropriate data, or ultimate question that one wants to answer. Okay, don't worry, we'll see these two things with the help of examples. Okay, it will be clear to you. And secondary objective is other thing you want to do along the way in clinical trial, means apart from apart from main goal, your second goal is your secondary objective. Okay, for example, this omeprazole 20 mg, I'm conducting one clinical trial. I'm conducting one phase 2 clinical trial. Okay, to evaluate the effect of omeprazole 20 mg in prevention of gastric ulcer. I'm running one phase 2 clinical trial. So you can see here, my primary purpose is to know the effect of this drug in the prevention of gastric ulcer. This is my primary objective. This is my primary goal. But along with this primary goal, I can do some other thing also, right? Anyway, I'm doing the clinical trial. I may have some other questions in my mind that I want the answers. So my secondary objective might be that I want to see whether my drug can prevent the lesions also in the esophagus or not. Whether my drug can show some effect in reducing the symptom of heartburn or not. Okay, so this might be my secondary objective, my secondary research questions. Okay, now let's see with the help of one more example. BI100073 is a drug that I am going to test to determine the safety and tolerability in patient with type 2 diabetes. This is my primary objective. This is my primary goal, a primary research question. But along with this, I can also determine the pharmacokinetic and pharmacodynamic of this drug. That might be my secondary objective, my secondary questions. Now, how I will come to know whether my primary objective is met or not, or my secondary objective is met or not? To determine whether my primary objective or secondary objectives are met or not, we test few things. Okay, what are those few things? Those are the things we call endpoints, primary endpoint and secondary endpoint. Okay, so for primary objective, whatever parameter we determine, those parameters are called primary endpoint. And for secondary objective, whatever parameter we determine, that is called secondary endpoint. Let's see the standard definition of endpoint, then we'll see. In clinical trial, an event or outcome that can be measured objectively to determine whether the intervention being studied is beneficial or not, that is called endpoint. Okay, now we'll see with the help of one example. For example, I have to see whether to access the adherence rate and effectiveness of 4 popular diet for the weight loss and cardiac risk factor reduction. So for this primary objective, this is my primary objective, right? I want to determine the adherence rate and the effectiveness of 4 popular diet for the weight loss and cardiac risk factor reduction. This is my primary objective. Now, how I will come to know whether my primary objective is met or not? For this, we check mean absolute change from the baseline weight at 1 year. Means we'll check the weight before enrolling the subject in a clinical trial, and after 1 year, we'll check the weight of the subjects. Okay. Then we'll see what is the percentage change. That is endpoint, means parameter. Now, weight is our endpoint here, right? Percentage change in weight is our endpoint. Now, understand, right? For example, um, of let's see one more example. We are testing a drug A, which is anti-diabetic drug in a phase 2 trial. Okay. Our primary objective is to know the effect of once daily dose of this drug on blood glucose level in diabetic subject. Okay. And our secondary objective or our secondary research question is what is the effect of this drug on overall quality of life. Okay, so here our primary endpoint would be how well the drug is going to lower the blood glucose level, like for example glucose lowering less than 120 mg per deciliter after 2 hours of dose administration. Okay, By measuring the blood glucose level after 2 hours of dose administration, we can see, right? We can see, right, whether the drug is really going to lower the blood glucose level or not. So lowering of blood glucose level after 2 hours of dose administration would be our endpoint. Endpoint is nothing but parameter. For example, if you are giving any exam, then that whether you are going to pass that exam or not, that is determined whether you have scored 33 or 40% marks in that particular exam paper or not. So that 40% or 33% is nothing but your endpoint. Okay, just understand in that way. Similarly, the parameter that we determine in order to say our secondary objective is met or not, what do we do? We measure few things. Okay, and whatever things we measure to say whether our secondary objective is met or not are called secondary endpoint. For example, for this particular example, what was our secondary objective? Our secondary objective was whether to know the drug A effect on overall quality of life. Okay, so we'll see the quality of life score, okay, after 6 months, in order to say whether our secondary objective is met or not. So you would have seen many press releases, right? Okay, this drug has met its primary endpoint. This drug has not met primary endpoint, but could not met its secondary endpoint. Okay, now you people can understand what they mean by this. So in real world scenario, to know the drug is successful or not in clinical trial is measured in terms of whether it met its primary endpoint or not. Because if drug is met its primary endpoint means the primary objective is successful, or the drug has achieved the primary objective, or its primary goal is achieved. Okay, now everybody is clear, right? What is primary objective, secondary objective, what is primary endpoint, what is secondary endpoint? Any questions so far? Then we can move to the next topic. Okay, so few additional point for the primary and secondary endpoint. So sorry, so the primary endpoint of a clinical trial is the endpoint for which subjects are enrolled and for which trial is powered. When I say trial is powered, what do you mean by trial is powered? Trial is powered means the whole trial is designed for the primary objective only. See, secondary objectives are our additional questions, right? The whole trial cannot be designed for secondary objective. The trial generally is designed for primary objective only, for the primary endpoints only. And secondary endpoints are additional event of interest. Okay, so always remember that trial always generally means, whenever I am saying always, it means, see, any sponsor company wants to answer want to get answer for n number of questions, right? But they cannot design trial in order to get answer for all their questions. They have to take few things in priority, right? This is my primary objective and this is my primary endpoint, and they have to design the whole trial according to those primary objective and primary endpoint. Yeah, by doing that, they can also try to answer few additional questions. Okay, they can also satisfy secondary objective and secondary endpoint. So whenever they are designing a clinical trial, they have to take good amount of subject in a trial. That is called that amount, that number of subject in a trial, we call it sample size. Okay, we we learned in phase 1 less than 100 people are required. In phase 2, 20 to 80 people are required. In phase 3, up to 300 people are required. And sorry, in phase 3, 3000 people are required. Okay, so whether they have to recruit 60 people or 65 people or 70 people in phase 1, they will decide based on their primary question or their primary endpoint. Okay, if they are not able to determine the exact number. It generally it may happen also. Okay, then the trial is not properly designed. It will lack the statistical power. Okay, then the trial is poorly designed and it will not give us a proper result. Now we heard about primary endpoint, secondary endpoint. One more endpoint are there. That's called surrogate endpoint. Now we learn about the surrogate endpoint. So what is surrogate endpoint? Surrogate endpoint is used in place of primary endpoint. Okay, to speed up the approval process or prove that drug appears to be working. Surrogate endpoints are used in place of primary endpoint to speed up the approval process or prove that drug appears to be working. So why surrogate endpoints are used? Let's see with the help of one example. For example, we are developing a drug which is used to reduce the high blood sugar, high blood pressure or high blood sugar. Okay, and we are checking whether our drug is going to decrease the mortality in subjects with high blood sugar or not. Okay, but to check whether our drug is going to reduce the mortality or not, it will take years, right? Then we have to see how many subjects are alive and how many subjects are died, right? It will take years to know. We cannot we cannot have that primary objective, right? And we cannot have the primary endpoint. So what we'll see, in order to know whether our anti-diabetic drug is going to reduce the risk of death from diabetes or not, what do we see? We see the blood sugar level. Okay, whether our drug is going to reduce the blood sugar level or not, we can determine whether it is going to reduce the risk of death from diabetes or not. So instead of waiting subject will die someday, we can use some other endpoint, right? We can use some other parameter, and this practice and those parameters which are used instead of primary endpoint are called surrogate endpoint. Okay, so in this particular case, instead of waiting subject to die, we are checking the blood sugar level to know whether our drug, anti-diabetic drug, is going to reduce the risk of death from diabetes or not. Surrogate endpoint. Okay, so whenever the primary endpoints are unacceptable, for example, our drug is to reduce the stroke, but we cannot wait, right? We cannot wait in a clinical trial that subject will have the stroke, and then we'll determine whether our drug is effective in reducing the rate of stroke or not. So whenever the clinical outcome is too difficult to measure, okay, or unethical, okay, whether our drug is going to prevent the abortion or not, those kind of whether the the primary endpoint is unethical or difficult to see in a clinical trial setting, we use a surrogate endpoint. Okay, our the primary endpoint will take very long time to study. For example, our primary endpoint may be whether our drug can reduce the risk of cancer in next 2 decades or not. How we'll determine? Okay, it's very difficult, right? Instead of that, we can determine whether our drug is going to increase the antioxidative elements or an antioxidative state of the body or not. I'm just giving one simple example. Okay, so in this example, instead of waiting to that subjects are going to have the stroke, what we can do? We can see whether our drug is going to reduce the blood pressure or not, because we know that if drug is going to control the blood pressure, then definitely the risk of stroke will decrease. Okay, so instead of having a stroke as a primary endpoint, we can choose the reduction in blood pressure as our primary endpoint. So in this particular trial, the reduction in blood pressure would be our surrogate endpoint. We'll call the reduction in the blood pressure as surrogate endpoint. For example, in oncology studies, instead of waiting that subject is going to die from the disease, what we'll see the progression-free survival. How many month subject can survive without cancer cells or without any active disease. Okay, so instead of checking the overall survival, we see the progression-free survival means disease-free state. Okay, so overall survival would be our primary endpoint. Okay, but as it is unethical or it is not possible to check in the trial, we check the progression free survival like for 6 months and 8 months. Okay, for eighth month subject does not have any cancer cell in the body, then this kind of endpoint we can use in our clinical trial, and those kind of endpoint would be called surrogate endpoint. Everybody is clear so far, right? Okay, so few more things about the surrogate endpoint. So surrogate endpoints that have undergone this extensive testing are called validated surrogate endpoint, and these are the accepted by the FDA as the evidence of benefit, because in last 30-40 years we have used many surrogate endpoint instead of primary endpoint. Okay, now we have good data. Instead of these primary endpoint, we can use surrogate endpoint, and we can determine the actual efficacy of a drug molecule. Okay, those kind of surrogate endpoints which have been used for many years, we have enough data. Those kind of surrogate endpoint are called validated surrogate endpoint. Okay, surrogate endpoints are also called surrogate marker. Okay, one more thing, whenever few places instead of endpoint they will use one word, outcome measure. Okay, outcome measure. Okay, outcome measure, always remember this thing. So till now we learned about discovery, preclinical, phase 1A, phase 1B, phase second A, phase second B, primary objective, primary endpoint, secondary objective, secondary endpoint, and surrogate endpoint. Okay, so now we'll see the classification of clinical studies. Okay, so mainly there are two type of clinical studies: observational studies and interventional studies. So whatever we have learned so far, that is regarding interventional studies. Okay, now what do you mean by interventional studies? Intervention, before we learn about the interventional studies, first we'll see what is intervention. So any medicine, any treatment, any procedure, or any action which we take to prevent or treat any disease and improve health in any way, that is called intervention. Okay, we have a fever, we take paracetamol. Paracetamol is the intervention. Somebody is having high cholesterol, they take the atorvastatin. Atorvastatin is the intervention. Somebody is having a COVID vaccine to prevent the COVID, that vaccination is the intervention. So whenever we are intervening the natural course of a disease, that is called intervention. Okay. So whatever we have learned so far, phase 1A, phase 1B, phase 2, phase 3, phase 4, whatever we have learned so far, those clinical trials are nothing but interventional studies only. Always remember. Okay. Generally, people will get confused with this clinical trial word. What is clinical trial? Clinical trials are experiment which we do to determine the safety, efficacy of a drug or medical device or any surgery. Okay, those clinical trials are nothing but interventional studies only, nothing else. Okay, now you know, right, what is clinical trial, interventional study, and what is intervention. Now, apart from interventional studies only, we have other studies. Now, what are those studies? Those studies are called observational studies. Okay. In interventional studies, we are giving drug, right? We are We are trying to change the natural course of a disease, right? But in observational study, we do not do like that. We simply observe. Okay. So observational studies are not the scope of today's class, but I'll give you general overview. For example, to determine whether smoking caused the lung cancer or not, we recruit 100 people and we observe those people uh, for 10 years and see how many people who are smoking got the lung cancer, how many people who did not smoke are having the lung cancer. We will do that comparison. Okay, we do not give any drug, we do not perform any surgery, we do not do any kind of intervention. Those kind of studies where we simply observe are called observational studies. Okay, in observational study there is nothing like phase 1, phase 2, phase 3, phase 4. Those are not observational study. Phase 1, phase 2, phase 3 comes under interventional studies only. Okay, clinical trials are nothing but interventional studies. An intervention is nothing but anything that we do to change the natural course of a disease, whether we are giving a drug, whether we are inserting any medical device, we are using pacemaker, or we are performing any surgery, or we are giving any vaccine, taking vitamins, whatever we are doing, all those things come under intervention only. In phase 1, phase 2, phase 3, all those phases are for clinical trials, and clinical trials are interventional studies only. Now, there are few things, few terms that we use generally for the clinical trial. Okay, first we'll learn about those terms, then we'll try to design a clinical trial. Okay, so the first, and I think many of you might be knowing this term, placebo. Okay, so what is placebo? So placebo, as per the American Cancer Institute, placebo is a substance or any kind of treatment that looks just like a regular treatment or medicine, but it is not. It is actually an active look-alike treatment or substance. So in clinical trial, what do we do? What do we do? Apart from a treatment, we also use an active substance. An active substance will look like a plus, will look like a treatment only. Will look like a original medicine, but it will not have any effect. For example, if we are doing any trial for say for back pain, and we are testing the diclofenac, so people will think, okay, I'm taking the diclofenac, my pain should reduce. Okay, they will have that mind will trick, right? That mind will trick and the pain will reduce. Okay, so to minimize, to cancel out that mind trick, to minimize that effect, psychological effect, we use this placebo drug. For some subject we will give the placebo, for some subject we will give the actual treatment. Okay, then we'll try to measure uh, the real outcome. Okay, that's why we use the placebo in a clinical trial. When I'll teach you about the trial design, like the blinding, masking, randomization, a proper trial design, the treatment arms, then you will have a better understanding why this placebo is there. Okay, but for now you can understand that placebo is nothing but an active drug. Okay, it will not have any effect, but it will look like a drug without any effect. Okay, that is placebo. The second term that we will use going forward, that is active comparator. You understand, right? What is placebo? An active substance with no effect. Now, what is active comparator? This term we are going to use very much going forward. Okay. So for now, just understand the definition. What is active comparator? So you would, if you remember, in while teaching the phase 3, I said in phase 3, we also compare our treatment with respect to the standard treatment available in the market. Okay. For example, if I'm developing a drug for back pain, any painkiller, then everybody knows that diclofenac is the standard treatment for back pain available in the market. So in phase 3, I will try to compare my drug with diclofenac. Okay, that 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 standard treatment that we are going to compare in our clinical trial, that's called active comparator. Okay, we have placebo, inactive, no effect. Then we have active comparator. very active. Okay, it's a standard treatment. So active control is nothing but effective treatment, which we compare to the experimental treatment. Okay, drug is compared with gold standard for that particular disease. For example, I have given one example here, levodopa. Everybody knows that levodopa is a gold standard treatment for what? For Parkinson disease. If I am developing any drug for Parkinson disease, then definitely the regulatory authority and sponsor will wish to know whether my drug is better or equally effective to the like levodopa or not. Okay, so in our trial, if I'm comparing my drug with respect to the levodopa, then I will call levodopa as my active comparator. Okay, everybody got it right. What is placebo, active comparator? Any questions so far? Okay, then we move to the next term, that is treatment arm. What is treatment arm? This is a third term, okay, and it's very important to know. So the group or subgroup of a participant in a clinical trial that receive specific intervention or treatment and no intervention according to trials protocol. Okay, so see, definition may have some jargon. Okay, so it's better always to understand with the help of one example. Let's see, I'm conducting phase 3. Okay, this is my phase 3. This is my phase 3 trial. So in phase 3, what I'm doing, I'm okay comparing my drug. Okay, let's see, this is a phase 2, not phase 3. Okay, let's keep it more simple. It's a phase 2. I'm comparing my drug with respect to the placebo. This is my drug, and this is a placebo. So few subjects will be receiving the treatment, right? And few subjects will be receiving the placebo. I'm telling few subjects will be receiving drug and few subjects will be receiving placebo. I can also say one group has received the placebo and one group has received the treatment, right? I can say in this term also, right? Instead of telling that few subject is receiving the treatment and few subject is going to receive the placebo, I can say that one group in my trial is going to receive the treatment and one group in my trial is going to receive the placebo. Okay, now I can say that my trial is having two arms. One treatment arms or one group is going to receive the treatment, and one group or one treatment arm is going to receive the placebo. So now you understand, right? Treatment arms are nothing but groups in a clinical trial, which is going to receive different kind of treatment or intervention. They may receive placebo, they may receive treatment, they may receive active comparator, but they are different group receiving different kind of intervention. Everybody is clear, right? Everybody is clear. Then we'll do one simple exercise, then we can close it. Am I audible? Poonam, Swarna.
Yes, sir.
Yeah, any question from your side?
No, sir.
Okay, I will do one simple exercise of 2 minutes, then we can wrap up this call. Okay. For example, you can see your chat. I'm just writing there. For example, I'm conducting one study. Uh, my trial is having 4,000 people. Okay, and I'm testing my drug A with respect to insulin. It's an anti-diabetic drug. I'm testing with respect to the insulin, and I'm determining what. Okay, so uh, if anybody can tell me, it's phase one, phase two, or phase three trial. If I'm having 4,000 people and I am testing my drug, which is anti-diabetic drug with respect to the insulin, it will be phase one, phase two, or phase three. Anybody can tell? Swarna or Poonam? Anybody? Any guess? Okay, you are typing. Poonam, thank you for your reply. It is going to be phase three because we are testing 4,000 people, and we are okay. And my second question was regarding how many treatment arms it will have. So it will have two treatment arms because we are going to test the drug with respect to the insulin, right? Definitely, to we are going to have two groups, and one group we are going to give the drug, and another group we are going to give the insulin, and we are going to test it. Okay. So, Poonam and Swarna, this is going to be phase three because we are having 4,000 people, and it will have two treatment arms. Because one treatment arm will have the drug, and second treatment time will have the insulin. Okay, so don't worry. Like in actual classes, we'll have more such exercises, and you will have more clarity. Okay, this was a quick summary for today's demo class, but in actual classes, learn more we'll learn about each thing with the help of more examples, and okay, and we'll have more exercises. If you people are having any question, then we can wait for two or three more minutes. Otherwise, we can wrap up today's call. Okay. Okay, then thank you so much, Poonam and Sonna. Then we can wrap up today's call and have a great evening ahead.